Okay, well, let's get started. Welcome to everyone in the room and online. And online, if you put a message in the chat if you can't hear anything. Anyway, hopefully you can hear. So this is a SYNAPSE Seminar, our transdisciplinary seminar series, supported by the School of Culture, History and Language in the College of Asia and Pacific. And I'd like to start by acknowledging the traditional owners of the Ngambri and Ngunnawal groups, whose unceded land we're meeting on today. So it's a real pleasure to have Monika Karmin visiting VHL over the last couple of weeks and presenting a second seminar. So thank you very much for the two seminars. So Monika is at the Estonian Biocentre at the Institute of Genomics at the University of Tartu, and her research is on genetics, paleogeography, and demographic histories of modern human populations in Europe, South Asia, Ireland, Southeast Asia, and Oceania, I understand. And she coordinates the Oceanic and Southeast Asian Navigators project and is also part of an Estonian Roots project, a Centre of Excellence for Transdisciplinary Studies on Ethnogenesis and Cultural Diversity. So very much aligning with a lot of the things that we're interested in ECTI. And for today's talk, Monika is going to talk about paternal lineages and their role in understanding population history. So really lovely to have you, Monika, and thank you very much.
Okay, I start. Hello, everybody here in the room and those in the Zoom who I can't see. I'm very, very glad to have this opportunity to present some of our work. Also, I want to acknowledge everybody who's receiving me here, and thank you very much for all the kind invitations and also very interesting discussions that we have had. It's been very enriching to be here. I think most of all, I also want to acknowledge all the donors of all of our studies, which have been very many, but anybody who has been willing to contribute their samples, either biological samples to our DNA studies, or then now most recently we have done a language collection, and there's been many people involved in there. There's too many to name everybody. Okay, so a brief outline. I will do a little introduction from where I come from and of this background, and then just a little bit about the Y chromosome and the studies that we have done, so in the global phylogeny, and then about the effective population size, and then few regional views, one from here, more closer here, from island Southeast Asia, near Oceania, and the other one is where I come from Northeastern Europe. And then just, I hope I can have a small, just a small introduction to also those projects that we have been working with. One is finishing now and the other one starting. So Estonia, where it is, it's in Northeastern Europe, a small country close to Finland, bordering Russia. We are speaking Finno-Ugric languages in our country, and this is quite a unique language family. In Europe, there is 3 countries where those are spoken. And it's just, I'm showing this also because our research is much related to this linguistic background as well. And just now there was a Congress of International Finno-Ugric Researchers in Tartu. And for your amusement, just some comparison with Australia, that the size of Europe and Estonia. So Estonia is kind of the size of Tasmania. So you would know the scale that we are working on. And I come from Tartu University, which is one of the oldest universities in the Nordic country. When you would Google, you find it was founded during the Swedish time, of Swedish rule in Estonia. And it is the biggest university in our country and has medical faculty, philosophy, so a classical university. This is my institute. So I am from the Institute of Genomics, where we do lots of medical genetics, actually, because Estonian Biobank, which is a very unique collection in the biomedical field, is also part of our institute. So a big part of our institute works with personal medicine questions. And then we have a group, former Estonian Biocentre, which has been already since the end of 1990s, basically, been working in the human population history and all these kind of questions related to this field. So our senior colleagues, they were the ones starting with mitochondrial and Y chromosome research. So we have this long history behind us. And I work, so also in our institute, we have people working with modern DNA from the living people. And then there is now a very thriving and growing group of archaeogeneticists working with ancient DNA. We have our own ancient DNA lab, which is led by our professor Kristiina Tambets, who is a professor of archaeogenetics. But my own work has mostly been in the population genetics of modern people or with modern DNA. And just below here is just few pictures also showing what have been done. Like we can look at frequencies of lineages changing in time. We build phylogenies, do also networks. So that's kind of the methods, also very classical methods that have been used. And I'm not going to talk about the ancient DNA at the moment. So just going back very basics, we all have our cells, which and they are very different. Most of them have the DNA in them, or they have a nucleus, which has our nuclear DNA. And then they are usually, or very often, you see these chromosome pictures. So this is actually a specific type in a cell cycle when the chromosomes look like this. Most of the other time, they are more like this kind of a ball of a mess of DNA there. But there is also this specific small organelle, which is mitochondria, that has a different, also its own genome. And mitochondria, and then the tiniest chromosome in the human genome, Y chromosome, they have a very specific way of being transmitted through the generations. So it is different from the other parts of the genome. Okay. So the other parts of the genome, we get both from our both parents, half and half. So when our cells are formed, but the Y chromosome comes only through the paternal line. So it is transmitted from fathers to sons only, or the majority of the Y chromosome. I'm not going to too many details now, but so let's say the majority of the Y chromosome is passed down only through the paternal line. And then this mitochondrial DNA, which was the small piece of DNA from the organelle called mitochondria, this is passed only down from mothers to the children. So men also have mitochondrial DNA, but they don't pass it on to the next generation. So therefore, we can trace maternal lineages with a mitochondria and paternal lineages with a Y chromosome. And when we think about the rest of the genome, this one goes through recombination in each generation. So the genome is mixed and shuffled, whereas the mitochondria and Y chromosome, these are passed on as single units, which means only mutations which accumulate through time are making up the diversity. And this is just very handy when you want to do phylogenies, whereas for the rest of the genome, it's just much more complicated. And each person's autosomal genome carries the whole population history, basically, whereas the mitochondria and Y chromosome would carry and paternal lineage history. Now we're back to the problem slide. So just again, this picture of the chromosome, and to show the Y chromosome, which I have been studied for quite a while now, and it is the most small, like the tiniest chromosome in the whole human genome. It is carried by men only, and because of this transmission from father to son, the phylogenetic tree, which we can see. So we can build a phylogenetic tree, which basically would encompass all the men in the world today. We could, if we had the data, we could do that. And this tree has like a nested monophyletic clusters. They are called haplogroups, lineages, clades. So these are used quite interchangeably in literature also. And they are named, have these alphanumeric names, which sometimes maybe whoever has read any mitochondrial or Y chromosome papers, you have seen these very, very long names. And this is just because of one of the naming conventions. These names do not have to be like this. And it becomes very, very complicated when you would add more data. And another way of naming the lineages is having this alphanumeric label and a marker label attached. Another complication there. So yeah, anybody doing Y chromosome work, I think naming and labeling is one of the biggest headaches of communicating this information, because we also do need to label them, because if we want to talk about them, then somehow we need some reference points. And when we have more data, we can also then calibrate those trees. So we would have split times for these clusters. So let's try again. Also about how the variation has been uncovered. Historically, a lot of data was acquired, and very great research has been done doing genotyping, which means looking at certain positions in Y chromosome that have been determined to be characteristic to a certain clade, and then this has been done in a large number of samples, and all the early research basically was based on this. And it was very, very fruitful. But this is still a limited amount of information, so you couldn't always differentiate between lineages. Therefore, you would have multiplication. So nowadays, what we can do, and this is what I have been doing in my research, is doing sequencing. Therefore, you don't look only in the certain positions, but you would look at the whole sequence underlying here. And when I say whole, it doesn't mean the whole Y chromosome. It's actually Y chromosome structure, underlying structure is very complicated. So there are just certain regions which have been accessible with a short read sequencing. But it's still long enough, especially compared to the genotyping data. So there is very much more information available now. It is about 10 megabases, which has been shown to be accessible with short read sequencing. And with this kind of additional data, then we would already know the number of mutations in those branches leading to nodes, which with mutation rate would give us time, and we can then have time calibrated and much higher resolution trees. So that is, I think, the main difference when we come from the genotyping studies and going to the sequencing studies, that we do the time calibration directly on those phylogenies. This is a very historic picture I like to keep, because I don't even know if it's up there in Wikipedia anymore, but this used to be in Wikipedia. And this is all research done in from 2000 to 2010, 15, basically. So all this was shown that the human Y chromosome tree, the deepest lineages are within, or deepest splits are within Africa. And we see those labels are A and B. So also, if somebody is doing also mitochondrial work, then in Y chromosome at some point of time, scientists came together and decided to relabel the whole tree more according to the underlying phylogeny. So it goes A, B, C. It's not completely like this, but it kind of goes that A, B, C, and those are the earliest splits. So within Africa, this did not happen in the mitochondrial world. So there it's the naming is according to how they appeared in the historical literature. So, and what was then discovered is that, yes, there are regional types, or every region has maybe some types of Y chromosome which are more frequent there, which does not mean that everybody there would have this. So it's always about the frequency of those lineages. And also you could see that some of the later or like New World, especially Americas, are much more nested within the other clusters, hinting to this population movements that and to their source populations. So what we have been doing already 10 years ago, time goes so fast. There was a study. Okay, so this was doing 8.8 megabases of chromosome-wise sequence from 459 men across the globe. So really sampling across the globe, trying to depict the global Y chromosome tree. And they were from 128 populations, and those have been shown all here on the map, sampling locations. And what we could see was also because in this study we had one lineage, one Y chromosome lineage, which is called A00. So this is a very early split. There is a whole interesting story related to how this lineage was found, because it's quite rare still. But with this lineage, the whole Y chromosome tree we dated was 254,000 years ago, as coalescing all male lineages. The rest of the tree is younger, and mostly what we saw was exactly what was known before, that African lineages are the deepest splits, earlier splits. And we also see that the non-African lineages, for all the Y chromosome lineages which are out of Africa, there is a short interval of time. We can always debate a little bit about the exact dates or numbers, because it's depending on the mutation rate, but the phylogeny is the same across different later another publication as well. So we see that there is this interval 52,000 years ago, when most of basically all the major lineages which are prevalent in outside of Africa today, they all rose at this time. Another characteristics is that in the last 15,000 years ago, there is this huge number of diversifications happening in across all the lineages in this Y chromosome tree, which probably thought is related to, well, it means that there was a population growth, and there was probably related to the climate improvement. And this it is happening after the late glacial maximum. And this has been shown in other papers by Bosnik, 2016, by Pille-Hallast. So at this time, there was like several big papers coming out, which were basically showing the similar story, just with a little bit different sampling. What we also did in the study was to look at the effective population size. So it is known that the men would have can have a smaller effective population size, which means people who are contributing their genetic material to the next generation. Because for a man, well, this is like a cartoon here, that if you take, there is a similar number of men and women here, I think it's 20 and 20, but it is possible that like all those women who are having children, and they are the ones contributing to the next generation, that it is possible that one man can be the father for all those children, whereas the others don't father any children. For women, because of the gestation period and giving birth, and it just takes more time, you just need more women in order to support this. So that is kind of the basic, very simple story. And so yeah, here it is 17 women, and 3 of them are who are having children, and 3 are not having any. And also, if we think now about Y chromosome, then Y chromosome is passed on only from father to son. So if one man has 100 daughters, your Y chromosome lineage, I'm sorry, is still dead. So that's kind of also part of the game. So always have to keep that in mind. In order to look at those changes in the effective population size, this is now the what's it called? Oh, I can't say. The plot, PSP plot, which summarizes those effective population sizes for Y chromosome across all those different, the same samples that we had, different from different geographical origins. And it goes from the past, which is here on the left side, and the present is where the 0 is. And we see that there is like a steady growth of effective population size everywhere, which then suddenly there is a huge dip in this population size. And when we compare this to the mitochondrial DNA from the same individuals, so these are men, so the mitochondria is from the same individuals, then we see that there is no such gap or dip in the mitochondrial effective population size. It just keeps growing as the population grows in the world, as we know. I remember during the time of analysis, and everybody was very suspicious about this, that that maybe it's an artifact, but it's also been shown in other papers now. There was at the time also a paper from Lipson, and also other papers have shown that yes, there is such a effect. And the time frame is about between 8 to 4,000 years ago. It's maybe not so visible from this graph, but basically it's not happening all at the exactly same time. So those different regions, geographical regions, have a little bit of a lag between them. So it's not like that they all go down exactly at the same point. We thought, what could it be? And if you could imagine that if it would all be at exactly the same time, then there would be like a pandemic killing men or something like this. But it wasn't like this. And also, just for comparison here, also I put up this comparing the mitochondrial and the Y chromosome images, because it's a bit misleading on this, putting those side by side. The mitochondrial effective population size has always been much bigger, and this is the biological reasons also behind it that I mentioned before. And then at this point, when the mitochondrial or the Y chromosome or male population effective population size is very low, the difference is almost 17 times between the sizes of male and female. So we hypothesized then during the time of the study that this could be something cultural. We didn't have any proof or anything. It's just a hypothesis which was based on this sequence of the timing of those Y chromosome bottlenecks going or happening in different regions, since at least by the knowledge at the time, it seemed to be following the establishment of already Neolithic, well-established Neolithic in different parts of the world, where you would already have a differentiation of wealth and power, therefore giving some male lineages maybe more opportunity to have more children in next generations. And this has to go through not just once, but it has to be passed on through the generations. So we would be able to see this. And yeah, then this is like the infographic from the time that I did this culturally driven increase in the variance of male fitness, which means some are giving more children to the generations and some less. And it's happening 4,000 to 8,000 years ago. Also, here is a different type of comparison between the mitochondrial and the Y chromosome effective population sizes. And then, of course, it sounded very good. This is the survival of the wealthiest, which was more like a newspaper style way of phrasing it. But it was just, that was what we just proposed, that one hypothesis. So time has gone on, and then there have been actually two very nice modeling studies which looked at this problem, because this usually people have very many different ideas why such thing can happen. So they would look at one of them was very strongly, or well, maybe not very strongly, but strongly for the violent competition between kin groups, and they modeled this. So this is from Markus Feldman's group. And then last year there was another study from France where they looked at the same data. They also had to have a very nice discussion about all the cultural knowledge that they had and from the ethnographic literature. And then they proposed that yes, the same effect can be also with a peaceful solution. So you wouldn't need to kill the others. So it's just a difference in the. So now jury is out still. And if I'm always asking if archaeologists can somehow provide us some kind of information which would, give the actual data from the fields somehow for either of those or some third hypothesis, because we just see this in the today's data sets. So now very quickly I will go through two views or maybe I will have to skip one because probably time is out. So to more local view here. So this is going back to that global Y chromosome phylogeny. And when we look at the lineages that are present from Southeast Asia or Oceania or island Southeast Asia, we saw on that tree that there are just very few of them. So the sampling was very poor. And the other studies, which were also based on 1,000 genomes, which is one of the public, very useful and very much used databases for population genetics, but they lack sampling from this region. So they also didn't help in this respect. And those are the main lineages that we see: haplogroup C, M, S, and O. So just to the labels. So we collected 380 samples from across this region. You see the sampling is very different from different locations. Some are maybe better represented and others quite poorly. But that was what we had available at the time. Basically, this is the result. Just to show you. I will walk you through this also a little bit, but so this is summarizing the results, also already more like trying to show the different time frames. So there's like zones there, 50 to 40,000 years ago, then 40 to 25,000 years ago, and so on as the colors go. And when we look at the early time, it is very, as we saw already in the global tree, but now we see more specifically in this region how all the major lineages, they arise during this period. So in all those haplogroups in C, M, and S, which are then considered the autochthonous haplogroups here, or lineages. But then what follows on the figure, these part where there's M and S and also some of those C lineages. So these are lineages that are mostly prevalent only in Papua New Guinea, in also very few samples we have, but from Australia and from island Southeast Asia. Next to this is haplogroup O, which is it's the most prevalent Y lineage in the world today, because it is in China the most prevalent lineage. So, and this well, it is thought that its origin is on the mainland East Asia. So something we see that within this lineage there is more splits happening in this same time period, whereas here, at least with our sampling, we have not been able to see much much new lineages arising during this period from 40 to 25,000 years ago. But then when we go on, now it's going back, and just to summarize it very quickly, and we can discuss this more detail later, that what we saw is that there is a pause, then around 25,000 years ago, there starts to be like a second wave of diversification within Highland Southeast Asia and near Oceanian lineages. Follows really like an explosion of activity, and then we already know that, which is also coinciding with the kind of the rest of the world's Y chromosome tree, where we saw that after 15,000 years ago there is much diversity arising, and then in Holocene there are different lineages present. So this is just another way. It's very busy slide, but here we just listed those different time slices within those different paternal lineages from the region. So in the circular tree, those yellow background is highlighting those lineages from the region, and then these colored dots here are showing just that within major lineages we would have those arising during these periods. So in Holocene, everything is happening everywhere. When 15,000 years ago, also we see basically many things happening in different lineages, but then we don't see any of the O lineages here in this region. And this is just another view to the same tree, but just showing how these very basal lineages, which are coming from between 50 and 40,000 years ago, they are really, some of them are in Philippines, some are at the moment with this sampling we have, we have only been able to sample in Wallacea, and then there is a number of Papuan lineages. So the colors correspond to the map on the side. So we see that in the whole region there is a very deep lineages present in those different, when we kind of categorize them differently into geographic groups. And just very few hints that we see on the tree, because sampling is very poor, especially from Australia. But what we saw that some of the Papuan and Australian paternal lineages have coalesencies 18,000 years ago, and others 3,600 years ago. And also much clearer, because this is much better sampling, we see. So this red should be the same kind of brownish color. We see how the Wallacean diversity is sitting within Papuan diversity, kind of, which has been then linked to back migration from or migration from Papua to these areas. And well, that's kind of the end of the story. We can see that I think the main, like for me, it's very amazing to see how we sample from, for example, Papua or the New Guinea island of New Guinea, and these guys cannot be geographically, they're quite close to each other, but their paternal lineage is like more or less 50,000 years ago. It's pretty amazing. It does also show our sampling, so it's not very comprehensive in this part, but still already much better than it was before. So, now with some more sampling from certain regions, we also start to see more recent events or more recent coalescences within those regions. So it is up to sampling, but still this island of New Guinea and in general in this region, we have this very deep lineages present. And so just to contrast this a little bit with the Northeastern European story, so this is from where I come from, again, back to this picture, Estonia and the Finno-Ugric languages. And because we've been speaking so much about the phylogeny, then I just point out that phylogenetically there is a lineage which is labeled N, and it has been shown to be very widely distributed as a whole, that this lineage as a whole. has this very wide distribution from far east in Kamchatka to basically Norwegian, northern Norway. When we zoom in, we see that in Finland and Estonia and Latvia, it is very highly prevalent. So the question might be that there is this East Eurasian origin Y chromosome lineage present in those populations. That when did it arrive, and could it be somehow related to the language that we speak, because our linguistic neighbors are more on the Eastern Eurasia. And you also would see here the difference in the countries, or like they are like political countries today, but geographically, when we move to Poland, for example, then this lineage is very scarcely present. So it is the blue part here, and the same in Sweden. So it is the really eastern border of this lineage is in Finland, Estonia, Latvia, Lithuania. So just pulling out the actual numbers for those countries, Finland actually has very high proportion, 60%, and Latvia, and Lithuania, Estonia are all in 30 to 40%. There is long history in our lab studying this lineage, because it's very dear to us. It is very present in our population, so we've been studying it for a long time. You just see another example here, how this going from this genotyping to sequencing changed the underlying picture or the information that we can actually get. So this was the info that was possible to have with the genotyping studies. But and it's the same tree now doing with sequencing. We see lots of substructure. This is a very Illume paper. It's a very interesting paper. We see suddenly lots of substructure, local lineages, but also maybe the main message was that there is one sub-lineage which has coalescence time around 5,000 years ago, and sub-branches of this lineage actually are distributed in the same wide area that the whole large tree was originally. So it meant, well, again, that somehow within the 5,000 years, these lineages have been able to travel quite far distances from each other. And then we come closer again to our home. We see that two of the sub-branches, which have been labeled N3A3 and N3A4, are specifically present, and they have like a contrasting frequency in Estonia and our southern neighbors and Finland. So N3A3 very present in the southern shore of Baltic Sea, and then the sister lineage on the northern shore. Very interesting. So we go on. We have a whole data set now of about 4,000 of these full sequences of Y chromosomes pulled from different sources. We are always trying to cover as much of our neighbors as possible. You might take a look at the numbers from Latvia and Lithuania. We are doing very badly there. So we cannot say anything actually about the comparison within this southern whole region. So while we have concentrated more between comparison between Estonia and Finland, because at least our samples are more comparable. And very quickly, what we see, so this is now 600 individuals on one tree. It's very hard to show those trees actually, but all the men today in Estonia who are carrying this lineage N3A3, their forefathers all have their common forefathers between 2,700 and 2,000 years ago. So a very much more recent times compared to what we saw, for example, in New Guinea. And the similar stories for the other lineage, where we have more Finnish presence. Still, the coalescence times are in the same time window or close to that. And again, we must say that populations which are south from us, we can't say anything about this at the moment because unfortunately the sampling is very poor. This is another lineage which is very prevalent in Estonia, also sampling from the same locations. And you see, for you to see the kind of the shape of the tree, how it looks, when you have so much more samples, but also that they still are coalescences are like very, so this is 10,000 here, so everything is below 5,000 years. And I just put the other tree that we saw before from regions or island Southeast Asia, near Oceania. So just to compare what we can see with this different sampling. So this is actually about 140 samples from this region, because it is just the lineage is called M and S, and then compare it to 300 or 936 samples from around Estonia, and the 40,000 years line is over here. We see here among those lineages much more branching happening, but it's also dependent again on our sampling, because here we are highly saturated to Estonian and Finland. So this is what we see in the tree. But haplogroup I actually looks like this pretty much. It's a very strange lineage. It has this very late expansion. So that was the end of the story from the Y chromosomes. And I will briefly go through this just to show you the two examples of two projects that we have, which are transdisciplinary and multidisciplinary, however you like to phrase it. So this is the Oceanic and Southeast Asian Navigators project, which brought me here. I'm very grateful for that, for this opportunity. And this project tries to bring together archaeology, biological anthropology, genetics, linguistics, and do joint data analysis. That was the original idea. The main, I guess, the problem which was faced was that there is already quite a number of genetic data available from the region of island Southeast Asia and Oceania, but the linguistic data was not comparable. At least that was the idea behind this project. So a new word list was compiled, which was over 12,000 words. And the idea was also to be able to have semantic fields which would show contacts between languages. And this was all done by Maren Klamer and Owen Edwards in the University of Leiden. And yeah, the other seminar was more about this. So I'm just going quickly through this. And the main idea behind this was that in the EU partners, so the project itself has EU partners and then partners outside of EU, altogether 26 partnering institutions. So this is how we are partners in this project. But the main idea was that EU partners would provide training for people in Indonesia and Philippines and Papua New Guinea to then go and collect the linguistic data. Some of them were already linguists in the universities. Some were affiliated field workers to Ifugao State University who were cultural workers in the Philippines and actually community members. Altogether, a number of workshops was done, so in those different fields, and eventually the data was collected and also analyzed, or the data curation was then done together. So it was two big events. First training event was in Uppsala University for 3 months. People came there. The next one was last year, 2 months in Tartu University. And there we saw also very important how it was to have the more senior academics from our Indonesian partners to join, because they could really contribute to the novices who had been just learning this and help them with data curation. So that, and I'm just glossing over everything. So this is the location of the sampling in the end. It did concentrate on the Philippines mostly, and then some data points also from Indonesia, and the comparative data set of linguistics and genetics And this we can do for the Philippines because the communities that were visited for the linguistic data collection, they are the same, which already previously there was an existing network for genetic study led by Max Larena from Uppsala University. So he is the main contributor there. And another very recent development is our new database. So for archaeology, this is a database of data sources. So it is not a you can, and everybody can contribute there if you know some data source which is missing or could be added. So it is listing the available archaeological open source data sets. Also open source, just differentiating between completely open, embargoed, or failing to actually retrieve it. But they are there and they are clickable on the map. And Sebastian Plutniak, who was here also with the auction secondment just a month ago or so, so he is behind this. It is a very new thing. And then jumping to Estonia, this is, I'm just telling that from 2017 in our university, linguists, archaeologists, geneticists, they formed this collegium of transdisciplinary studies. And I think this kind of having such an umbrella organization or somehow coming together and then also contributing some funds, but it was more just to have a structure to come together. And they developed courses. So every year they have one course, which all of those fields are covered which is provided in the university. And this has become very fruitful in this sense that this really brought people together and made them talk to each other. And also, of course, when you have a commitment to have a course in the university, you always are more committed, I guess. And the result of this has been this online resource, which is just summarizing all about Estonian history and also in English it's available. So this is a start of this more formalized transdisciplinary studies in our university. And then last year, or actually the application was already before that, but there is a new Center of Excellence, which Beth, you read so nicely. I'm having always trouble wording it out. So this is a Center of Excellence bringing together now even more fields and 3 different institutions. So it is also having Estonian Literary Museum and Tallinn Technical University. And it is bringing together paleoclimate, linguistics, archaeochemistry, archaeology, archaeozoology, archaeogenetics, folkloristics, and cultural geography. So all those fields in order to give a better picture of the population history and also relationship to the environment in our region. And this is just now started. So it will be 7, it was like, it is Center of Excellence. So it is 7 years of funding. And it's now the 2nd year. And I think the challenges should be bigger, made like a bigger font here, because these are really the main challenges are the finding this common language between the disciplines, and it takes a long time. And luckily this project actually it was planned in there that we sit down and we start to really go back very, very basics and talk to each other about what's our data like and what are the questions. And then in those discussions, people go like, huh, what? And then you actually can ask, find this common ground and maybe ask some questions together. And most importantly, know that, okay, this cannot be done. These questions I cannot answer with this method, which is very often, I think, at least for me, when I read other papers trying to put information just side by side, I always I wouldn't know all the limitations of the other field so well. So this is the idea here, and also the acknowledgement, and those disciplines do cover different time frames in the timeline, like linguistics and folkloristics would be very recent times that we can go back, but very interesting again. For the bioenvironment, of course, this is much longer history. And just to remind everybody here in the room that our place was under the ice during the last glacial maximum. So it was like 4 kilometers of ice on top of us. So all our landscapes are actually formed by the retrieving ice, and people could not, even if there was somebody there, we wouldn't have any trace of them. So that's somehow maybe our time frames are shorter and we can go more thinner slices. Just to compare to here, I'm always so amazed by the depth here in this region. Yes, and of course data incompatibility, this is just another topic we can talk a long time. So thank you very much. Sorry I went so long. This is our auction partners, so universities in EU and then all our overseas partners. And this list has been changing. There is some people from Tartu University, my colleagues. Pictures are quite old already, you never can keep up with those, but we're always very, very thankful. If any Y chromosome, like if you really are like deep interest, then Professor Siiri Rootsi and Richard Williams, they are the ones who are like the mother and father of Y chromosome study in our field. And the island Southeast Asia and Oceanian research was much supported by François Rico from France, who's also part of OCSEAN, and then Mari Cox from New Zealand. And Toomas Kivisilm is just kind of the head behind many, many things, what's happening in Tartu, because now Toomas is actually in Leuven University in Belgium. And Mait Vespalo is our director of our institute, but very supportive in all this. Thank you.